Hello, Pirates. It's Thursday. I hope you guys are having a wonderful week and, understanding membrane transport a little bit. The next topic is specifically osmosis. So, all molecules entering and exiting the cell are very important, but as you guys have kind of learned with the properties of water, this year, water is just extra super special. So it gets its own word, and that word is osmosis. So your quick notes on osmosis are going to go onto page 27 in your notebooks. And with osmosis, osmosis is all about the movement of water. So when we're talking about osmosis, the special type of membrane transport, it is the movement of water into and out of the cell. So it's all about the movement of water. So when you guys hear osmosis, you should automatically think water. And something you might remember about the movement of water and osmosis from our membrane transport notes is that osmosis is a type of passive transport. So this is passive transport. And if you guys recall, passive transport required no energy. So it did not use any of that special molecule ATP to move water from one side of the cell membrane to another. And passive transport is going with the concentration gradient. So it's going to move water from areas of high water concentrations, so high H2O, to areas with lower water concentrations, so lower H2O. All right, so when we're thinking about osmosis, in your diagram here, you guys want to just think about a cell, the average cell. And so the average cell in a living thing, so this little circle here is just a cell, but the typical average cell is about 70% water. So somewhere around like 65 to 70% water. So for our purposes today, we're going to say 70%. So they're about 70% water, and they're about 30% other stuff that's dissolved in the water. So we call that other stuff a solute. So the stuff dissolved in water is a solute, and that makes water the solvent. And if you guys remember, water is the universal solvent. So the cell on the inside, so inside the cell, the cytoplasm, is about 70% water and 30% solutes. So when we're looking at osmosis, the water is going to move from areas that there is a higher concentration of water to an area where there is a lower concentration of water. So if we look at the very first scenario, so I'm just going to zoom into scenario 1 here. Here we have our beaker. And so in our beaker is 20% water, and it is 80% solutes. Okay, so here we have 20% water, so 20% H2O, and it is 80% the solutes. Okay, I'm going to put that right there. So when we look at this, our cell is 70% water on the inside and 30% solutes. So our water is the one we're interested in. So the water is going to move from an area of high water concentration to low water concentration. So what you want to look at is there a higher concentration of water inside the cell or outside of the cell? And 70% is higher than 20%. So here we have high water and low water by comparison. So the water is going to move out of the cell. All right, so 20% water in the beaker means is an 80% solutes in the beaker. And then our cell, 30% solutes, 70% water, 70% higher than 20%, water moves out of our cell. What happens? Over time, the water exits the cell. And so what's going to happen to our cell, and think of like a water balloon. If I have a water balloon and the water leaves my water balloon, then my water balloon is going to shrink. So if it's an animal cell, the cell is going to shrink. And if I'm in a plant, the plant cell is going to wilt. So the plant cell will wilt when it loses its water. And so what happens to my animal cell, I'm just going to use a red blood cell for example. It's usually a nice red round circle like an inner tube, but what's going to happen, it's going to kind of start to shrivel up as the water moves out of it. So the water moves out and the cell shrinks. In my plant cell, the central vacuole loses water and my cell starts to collapse. So here's my little central vacuole and the water goes out. For this solution, we say that a solution like this is hypertonic. So this is referred to as a hypertonic solution. So hypertonic, that prefix there for hypertonic, it means over or higher. And in this scenario, the hypertonic solution, we have a higher water concentration inside the cell. So the water concentration is higher inside of the cell, so the water moves out. For the solution has a higher solute concentration, so the water moves out to balance it out because it's always trying to keep everything at equilibrium. All right, so this is a hypertonic solution. The cell is going to shrink. And our next scenario, so scenario 2. Scenario 2 says that we have 70% water in the beaker. So I've got 70% H2O in the beaker, and I've got 70% water in the cell. What's going on here is they are exactly the same. Now that doesn't mean water is not going to move across the membrane, it's just going to mean that it moves across equally. So the water is going to move equally between the inside of the cell and the outside of the cell. So water moves back and forth equally. We say that this has reached a state of homeostasis. And homeostasis means that we have the same state. So here's my red blood cell looking normal and healthy and full. And the water is just going to move in and out equally. And then for my plant cell is going to be healthy and full. And so the central vacuole is going to be nice and full of water. And so we call this kind of solution that the cell has been placed in, this is considered to be an isotonic solution. So it is isotonic. An isotonic, it means that it is the same. So iso means the same, like an isosceles triangle. Or do they have the same side length or the same angles? No, you guys are in geometry. You tell me. So the water is equal and they move back and forth equally. Or if you do like an isometric hold for fitness, you're holding the same position to work your muscles. So isotonic is the same, homeostasis, everything remains equal. All right, our last scenario. So we've had water is higher inside and lower outside. Water is the same. So our last scenario, we're going to have a high amount of water on the outside. So this very last scenario, we've got 90% water on the outside of the cell. And we have our basic cell, 70% of water inside the cell. So in this scenario, there's a higher concentration of the water outside of the cell compared to inside. So the water is going to move from high water concentrations to low water concentration. And over time, water is going to move into the cell. So water enters the cell. Now for an animal cell does not have a cell wall to give it structure. So what can happen to our animal cell is it can fill with so much water that the cell will actually burst open. So this cell will swell, and it could actually burst. It could potentially burst. In a plant cell, the plant cell is going to fill up and it's going to put a lot of pressure on the plant, and they call that turgid pressure. So we have the central vacuole, and the central vacuole is going to put a lot of pressure on the cell wall. And we call this kind of solution hypotonic. So this is turgid pressure. And so this is a hypotonic solution. And the prefix here for hypotonic, hypotonic means under or lower. And this means that we have a low water concentration inside the cell. So the water concentration inside the cell is lower than outside the cell. All right. Now, as you start thinking about osmosis and thinking about water moving, think of how this affects your hydration. Think about how this might affect a fish. What if you got a saltwater fish and you put it in a freshwater tank? What would happen? Well, there would be a higher water concentration in the fresh water than in the fish. And so what would happen is its blood cells would shrink. If you put your saltwater fish into a freshwater tank, the blood cells in the little fish will start to swell, and some of the cells could burst and kill the fish. So with this, the goal is to have isotonic solutions so that everything stays at homeostasis and balanced. This is why IVs you get at the hospital are trying to help achieve homeostasis. They've got a balance of things like sodium, potassium. Think about if you drank too much water. Could you drink too much water? Is it possible for that to be unhealthy? And then think about something like Gatorade. Gatorade is a goal at creating a more isotonic solution for athletes. Athletes need to make sure they're replacing not just the water, but the solutes as well. All right, what you guys want to do is put this on page 27, watch the videos about osmosis, and then if you have a gummy bear or a raisin, put it into water overnight, just a pure water solution, and see what happens. I will see you tomorrow. Friday, you can log in at noon. I'm going to put some gummy bears in water here on Thursday, and you guys can see what's going to happen to them overnight, and you'll get data and some osmosis practice. So I will talk to you later. Have a good one.